Welcome to the lecture 4 of the lecture series on the enzymes used in recombinant DNA technology. Today's topic it is the polynucleotide kinases. The kinases are the enzymes which are involved into the phosphorylation. Here, the source of the enzyme it is a bacteriophage T4 infected E. coli cells. That is, from the E. coli cells there is a extraction of the kinases, and that kinases they are synthesized by the T4 bacteriophage. So when a bacterial cell it is infected by the T4 bacteriophages, it is going to synthesize the polynucleotide kinases, and that synthesized polynucleotide kinases which are present into the E. coli that it is going to be extracted. That's why the source of the polynucleotide kinases it is a bacteriophage T4 infected E. coli cells. Next, the substrate, it is the single stranded DNA or it may be a double stranded DNA. But the requirement it is what? It should have the 5' OH. Second substrate, it is the RNA with the 5' OH. Third substrate, it is a DNA with the 5' phosphate. Next here, the reactions which are going to be catalyzed by the enzyme, it is a forward reaction, exchange reaction and a 3' phosphatase activity. Now, in some of the cases, what is going to be happen at the 3' end, instead of the OH, there is a presence of the phosphate. That phosphate it is going to be removed with the help of the kinases. Okay, so here there is a 3' phosphatase activity. Okay, that is a removal of the phosphate group from the 3' OH. That is a 3' phosphatase activity. Now going towards the first reaction, and that it is a forward reaction. In this forward reaction, the substrate which is going to be there, it is going to be a DNA or RNA which is having the 5' OH. And here the donor of the phosphate, it is a ATP. Okay, it is whom? It is a ATP. And the here from the ATP, the phosphate which is placed at the gamma position, it is going to be donated to the DNA or the RNA. Okay, so the bacteriophage T4 polynucleotide kinases catalyzes the reaction of the transfer of gamma phosphate of ATP to the 5' OH of the polynucleotide chain, that is the DNA or RNA. Now here you can see this reaction, DNA it is with the 5' OH, RNA it is with the 5' OH. And here the enzyme it is a bacteriophage T4 polynucleotide kinases, and here cofactor it is a magnesium ions, and here there is a use of what? There is a use of the ATP. Okay, so the gamma position phosphate now it is transferred to the DNA and RNA. Now here you can see that DNA it is with the 5' phosphate, or the RNA it is with the 5' phosphate, and the byproduct it is going to be a ADP. Second reaction, it is an exchange reaction. Okay, as the name suggests, there is a give and take reaction. So here in this exchange reaction, in the presence of the excess, okay, the word it is what? Excess ADP causes transfer of the 5' phosphate of the DNA to ADP to convert, okay, to convert ADP to ATP. Okay, what is going to be happen? The ADP, it is going to be converted into the ATP, and now here the donor of the phosphate, it is whom? It is going to be a DNA. Okay, it is going to be a DNA, and the 5' phosphate of the DNA, it is donated to the ATP when the concentration of the ATP in a reaction, it is a higher one. Okay. Now here in this reaction you can see that this it is a polynucleotide chain, and here there is a presence of the phosphate at the 5' end, and here it is an excess of ADP. Now when you are making the reaction mixture, in the reaction mixture you are going to adding the bacteriophage T4 polynucleotide kinases, and here you are going to add the DATP, okay, and here you are going to add the magnesium as a cofactor. Okay, so as the excess of ADP is there, that's why the phosphate it is donated to whom? It is going to be donated to the ADP. ADP it is going to be converted into the ATP. Now here in the reaction you have added what? You have added the DATP. So this DATP, ATP now it is going to donate its gamma position phosphate. Okay, it is going to donate the gamma position phosphate to whom? To the 5' end. So now here you can see that the 5' phosphate it is going to be present. So this reaction it is going to be called as an exchange reaction, and this exchange reaction most of the time it is used for the labeling of the radio labeling of the DNA with the help of the phosphate. That's why here it has been written the gamma okay, a gamma position phosphate, which is of the 32, that is a p32 label, it is going to be a there. So here the star it has been denoted. Why? Because it is a radio labeled now. Okay, so in this way there are the reactions which are going to be carried out by the polynucleotide kinases. It is a forward reaction, exchange reaction, or it is going to be a 3' phosphatase activity. Now here going towards the applications. Applications, there is what? Radio labeling of the 5' termini for the Maxim and Gilbert DNA sequencing. Then you can do the radio labeling with in the nucleus S1 analysis, in the preparation, and you can go for the phosphorylation of the 5' termini of the synthetic linkers and the DNA fragments. Okay, during the cloning, when a foreign DNA molecule you have to clone into a vector molecule, at that time there is a removal of the phosphate. Okay, to avoid what? To avoid the self-ligation of an vector molecule. Now the ends of the vector DNA, it is now how they are consisting of the 5' OH. As there is a dephosphorylation with the help of the enzyme, and that enzyme it is alkaline phosphatases. But when you have to, when you have to ligate your foreign DNA or a gene of interest into a vector molecule, at that time the ligation it has to be done. And the condition for the ligation, it is what? There should be a 5' phosphate. But by the alkaline phosphatases, to prevent the self-ligation, you have removed the phosphate group from the 5' end and generating the 5' OH. Now, for the ligation, there has to be the 5' phosphate, and for that purpose, there is a use of what? There is a use of these polynucleotide kinases, which is allowing the conversion, or which is adding the phosphate group at a 5' end. Thus, allowing the ligases to act on the DNA, so that the vector DNA now it is going to be sealed with the gene of interest or the foreign DNA. In this way, your polynucleotide kinases it is going to show its application. I think you may have cleared the idea about the polynucleotide kinases. If you like my videos, please subscribe my channel and also share my video with your friends so the knowledge it is going to spread. Thank you.